tests, and also some policy recommendation. This will become more clear in the following time of this webinar. Next, please. Now I would like to introduce you to the biodegradation team of ReBIOlution. It's on one side, on the left side, Stephen from Normec OWS. He working mainly on the composting topic, although also very experienced in soil and aquatic testing. And then we have in the middle Michael Sander from ETH Zurich, very experienced in soil environment and testing and focusing on research questions. Ourselves from HYDRA, we are our expertise mainly in the aquatic environment, and we will share our insights then later today. Next, please. So I would like, before handing over then to my colleagues, give a little bit of a framework why we are doing this, what's the motivation of looking at biodegradation in such a big project where new materials is developed. We have identified with a group of experts working for the chief scientific advisors of the European Commission. You see the reference on the bottom of the slide. And we have identified 3 main categories where we think is very useful to substitute with biodegradable polymers. On the left side, we have the intentional release to the environment, and recovery is not foreseen or impossible. One example is the mulch film, as displayed here in a horticulture of strawberries, or in a more engineered and managed environment like the compost, here as an example of fruit stickers ending up in compost and potentially then polluting the compost if this material is not biodegradable. Category number 2, in the middle of the slide, high potential of loss to the environment, recovery again often impossible or not feasible. Here you see in very similar color between the sticks some plastic pieces of a vineyard area where they have some clips to actually hold the plant to their wires, and often they are lost to the environment and no one is going to pick them up. So very useful to have here biodegradable material in soil environments. Third category, unavoidable release into the environment, recovery is impossible. Here we have many examples. One here in the picture should simulate like a wet wipe in an aquatic environment, losing a lot of fibers when a little bit shaked around. These fibers are impossible to be recovered. If they are from plastic, persistent plastic, they will just simply remain and accumulate in the environment. Another example is here, maybe you see it here in my screen, is a brush that on one side, the brush is much shorter than on the other side. So by just simply using this brush on a hard surface, we are abrading a material into the environment in very fine grains or dust, which are then also accumulating in the environment. So there are many motivations, and the list is much longer than the examples I have compiled here. Just look around yourself in your environment where you have input to the environment you cannot recover. Next slide, please. So how to assess biodegradability and biodegradation time to actually then find a framework how to regulate it and put it into policy. We have different topics to address. On the left side in this block, here we address the tiered test approach that you want to have first the proof of biodegradability under optimized conditions. Usually it's done in a closed flask. So that you can capture an end product like CO2, like of the mineralization process, and you want to have optimized conditions because the test should then be quite feasible and quickly done, that you have the result that your material is fully biodegradable. However, this is an issue and heavily discussed also among stakeholders, that this is not representing then the real environment, nevertheless not a product. So usually in this tiered test approach, you then assess disintegration time as a proxy for biodegradation time of the product under more relevant conditions. We will hear then later also in a more practical way how that is meant. And the third level is then to prove minimal harm, simply done by ecotox tests, usually on 3 levels. You have like a phototroph, an algae tested with the material you have exposed in the biodegradation test, and then an invertebrate as a consumer, like a copepod or a daphnia, and also a decomposer, which are microbes. So you have the 3 main trophic levels assessed on ecotox testing. Now you see here 2 more boxes to illustrate and explain that we have a standard test view and a research-focused view on how to assess biodegradation. To make it actually comparable, usually standard tests should be used. These are established tests and validated tests. You have a pass or fail criteria, usually at a certain time frame you have to achieve, or a certain amount of biodegradation, maybe in percent or another unit, and they're easy to compare. However, they will not cover all the questions we have in this respect, so it's very important also to have research-focused assessments and options, methodologies to be used. However, they are not bound to standards, so you can also use other methods. We will learn in the webinar from Michael, very interesting methods to be used in this respect, and usually we do not have a pass level to fulfill, so they are open to a time frame, which is also quite interesting to look at, especially in the development of new tests than being implemented in standard tests. Potentially not easy to compare, that's the contrary compared to standard test. Thank you. Next slide. In terms of policy, we decided to go through 3 steps to give it a framework here in terms of biodegradation. And the European Commission is acknowledging biodegradation and sees it as a part of a broader integrated approach to waste management, recycling, and sustainability. So we cannot look at it as biodegradation alone. We have to think about it in a more broader aspect. Here, sustainability is meant by in the footnote, you can see that it's relating to sustainable and safe by design. So the projects of the European Union and also of the European Commission and also of the research projects are meant to actually following this thinking that a framework for the sustainable and safe by design requires the integration of risk assessment and safety-based considerations with life cycle-based considerations to ensure sustainable along the entire value chain. And the focus is always in respect of looking at the waste hierarchy. So first it should be prevented. We should reduce the use of plastics. We should promote recycling and reuse, and then potentially redesign according to sustainable, safe, and safe by design, with biodegradability as an option, but under certain conditions, and with very clear labeling and communication. Focusing on added value and benefit. This is clearly communicated in the EU policy framework on bio-based, biodegradable and compostable plastics. So we have to look out for when does it really make sense and what certain conditions are meant and how do we have to assess them that we are complying with a policy in place. Next, please. Where is it currently implemented? It's actually, you can say it's under the Green Deal, the Circular Economy Action Plan, and also the Zero Pollution Action Plan. So it's fueling down into more clear policies than be it under the waste management policies or plastic product policies, all guided by the EU policy framework here. The first bullet point, which is the framework addressing the role of biodegradable plastics in reducing the environmental impact of traditional plastics, which are accumulating persistent plastic in the environment. So this should be reduced and worked on. We have to reduce this kind of pollution in the future and as soon as possible. We have 3 regulations in place where we have indication and clear guidance how to work with biodegradable plastics. In the packaging and packaging waste regulation, for example, is considered biodegradable packaging as one of the potential solutions to reduce packaging waste and environmental impact, but it does not prioritize biodegradation as the only solution and emphasizes that biodegradable packaging materials must be properly disposed of in composting or similar systems, ensuring they do not contribute to pollution or complicate the recycling efforts. So again, you see here this integrated approach, which has to be put into frame. In the REACH regulation recently was clearly adopted that restrictions on microplastics used in consumer products. So this is not very directly related to our project. However, we can use it as a guidance, and because the European Commission will not much deviate from that, it says that biodegradable products can be exempt from certain restrictions if they meet specific biodegradability criteria following standard tests. So you see here the standard tests are a very important instrument and the criteria to be defined then in these specific regulations. Very interesting, last year, fertilizer product regulation, the last bullet point, which is the first regulation where biodegradation is implemented as a must. Why? Plastic in agricultural and horticultural products, such as controlled release fertilizers, for example, here, I hope you can see these green dots are very, very small slow release fertilizer granules made partly out of persistent plastics, so no one will ever collect this again out of the soils. So the Commission decided that we have to change here, we have to substitute, and these plastic materials have to meet specific biodegradation criteria, again following standards, to ensure that they break down safely in soil and minimize their environmental impact. Also implemented the mulch film in this regulation. So these parts, when they are plowed under in the soil, have also to biodegrade in a certain amount of time. Thank you. Next please. How is it done? The implementation into policy, it's looked at where do we have added value, mostly in the reduction of pollution, some also in the compost area in increasing the collection of green waste, and referring to standards and that you should do the tests by accredited labs so that you can show independent test results by, for example, in the REACH regulation, by so-called OECD tests, or the packaging and packaging waste regulation. In terms of composting, you should demonstrate biodegradability by using EN ISO or national standard tests. Stephen will give more details on that. And you can also do some testing in open environments. However, this is not promoted in this regulation. But it is a must in the fertilizer product regulation, as said before, and you should not only test in soil, you should also test in water for the option that this mulch film is flushed into a nearby water environment. All around is, again, the policy framework giving the structure how to implement in the regulations the topic of biodegradation. It always emphasizes proper testing and labeling so that no misunderstanding can occur. And it also identified some gaps, some missing tests in relation to biodegradation, for example, during transfer. So when from compost to soil, plastic is transferred, or from soil to water, and maybe then eventually even ending up in the deep sea. I will go in more detail later in my part later on. Next, please. So this leads us now to the three environments we're looking at in the project of ReBIOlution, the composting or the compost environment. On the left side, you have the home composting, and on the right side, the industrial composting environment. In the middle, there's a picture of a horticulture where the culture had been changed, and some of the mulch film has then been ripped off. In the lower part of the picture, you see a mulch film laying on the surface, and so kind of releasing or lost into the environment in a soil environment, and eventually flushing into the nearby water here in the aquatic environment on the right side. That gives you an idea of which environments and why we are looking at engineered and open environments. Next, please. Leading to Normek OVS. Stephen, this is your part.
Thank you, Miriam, for this nice introduction. So my name is Steven Verstichel, working at Normek OWS. Just some few things on our company. We are located in Ghent, Belgium. Also have a daughter lab in Dayton, Ohio, in the United States. We offer services in four main pillars. First of all is on biodegradation testing, compostability testing, and ecotoxicity testing. This is so not only for compost, but also in soil, water, and marine environment. And for this, we are recognized by all certification institutes worldwide. We're also strong believers in standardization, so that's why we are a member of the standardization committees in the field of biodegradation and composting. Apart from that, we offer waste inspection, so we could check the quality and of the sorting of industrial installations on plastics, but also on, for glass, etc. We have a sustainability assessment department where we offer life cycle analysis services, and as last, we have some consulting services like this kind of webinars and workshops we organize. Next slide, please. So composting, what is actually composting? You can see here the definition of the European Environmental Agency. So it's the controlled biological decomposition of organic material in the presence of air to form a humus-like material. So our start material is organic material. As you can see from the picture, it's wood waste, grass, clippings, leaves, yard trimmings. And in the presence of oxygen and microorganisms, this is converted into compost. Compost, which can be done as a soil improver, will regulate in better nutrient management and reduce soil erosion. So we have a higher plant growth and a better soils. From the picture, you already can see that there's some plastic inside. So we have a lot of food packaging, which would be relevant to make them also compostable. But how should we treat this packaging, and which requirements should they fulfill? That is all defined in the European standard EN 13432, and it's really sets the requirements for this packaging, these plastics, these paper products to be allowed to go into the organic material fraction, but that they will not serve a negative effect on the compost process and on the final compost quality. So we want to ensure that our final product is really of a high quality and can be used to improve the soil. Next slide, please. The standard in this EN 13432 is really related to industrial composting conditions. So in this installation, you have really high temperatures above 60 degrees Celsius for 1 week, and then the temperature profile already slows down. It is quite similar like other standards, like in the United States, you have the ASTM D6400, in Australia, the AS 7436, and on international level, the ISO 17888. Mainly, you have 4 requirements that have to be fulfilled: 2 for environmental safety and 2 on degradation. So on environmental safety, you should not have heavy metals in your product. You should have enough organic material, as your plastic is also organic material. Should be high enough. We want to compost organic material, not stones or inert materials. Furthermore, it should have no impact on the composting process, and finally also on the compost quality, which is also proven by ecotoxicity testing, and in this European standard mainly by plants. But apart from this, your material should also degrade, should be composted. So it should degrade under the composting process, which is checked by 2 tests. First one is the biodegradation. It's really the mineralization of the carbon in your material, in your plastic, in your paper, into CO2 and water, and actually some biomass. And the other one is the disintegration, which means that your material, your final product, must fall apart during the composting process, and it should not be visibly disturbing your compost. So it should be smaller than 2 millimeters, that you're sure it's really can be treated by a composting process. Next slide. So if you go a little bit more in detail in this European standard EN 13432, it forms the basis of several certification schemes, like the OK Compost Industrial logo from TÜV Austria, Belgium, the seeding logo from European Bioplastics, and the DINC approved OK Industrial from the SATCO. And has the 4 requirements, as mentioned before. The first one is characterization. The test methods are prescribed there, but it's mainly the requirements for heavy metals should not be containing. There should be no fluoride inside, and you should have enough volatile solids. The second one is in biodegradation, where you should prove the biodegradation under controlled composting conditions, which is typically done in a test according to ISO 14855, where you have a maximum duration of 6 months, and your test item is then exposed to major compost, and the CO2 production is followed, so that we can determine the exact biodegradation. The requirement is that you should achieve 90% biodegradation on base of CO2 within 6 months. The third requirement is disintegration, where mainly the one as ISO 16929 is followed. And this is really your material must disappear, fall apart during composting process. And that is done by simulating an actual composting process. And the duration is a maximum of 12 weeks. And all your pieces should be smaller than 90% within this period. It should be more than 90% of the piece should be smaller than 2 millimeters within this period. And finally, we determine the compost quality, also not only by ecotoxicity, which is typically done with 2 plant tests, where you have to have achieved more than 90% germination and biomass compared to a control reactor, but also just parameters like nitrogen, phosphor, salt content are followed, and the maturity to really conclude that your product is not bad for the composting environment. Once you fulfill these requirements, you can put next, you can achieve several certificates, like I mentioned before. Yeah, you can push next. Yeah, you see there the 3 logos, and there on the right you see the one from BPI, which is from America, which is still based on the American standard, and the last one is from Australia and the Asian region. Okay, next slide. So this is really for industrial composting, but in home composting, these high temperatures are often not reached. So that's why we need to have other specification. In your home composting process, you will not reach the 60 degrees Celsius. We will reach a maximum temperature around 25, 30%, a little bit above the environment. Therefore, the biodegradation requirement is testing is done at 28 degrees, so not at 58. And you should achieve 90% biodegradation in 1 year. So it's a longer period also. Similar for disintegration testing, you should test it at 28 degrees, and you achieve that more than 90% of the material is smaller than 2 meters within 26 weeks. The requirements for chemical characteristics and ecotoxicity are similar to the one as for industrial composting. Several standards are existing there. One that is often used is the French standard, also the NFT 51800. But the first one is the European standard for carrier bags, and then you have also the Australian standard for home composting. And once you achieve fill these requirements, you can also have different logos as seen below. From TUS Austria, Belgium, you have the OK Compost Home. This has to go the Home Compost approved, and also the one from Australia is mentioned here. Next slide, please. What is important that is now also more recognized by the European regulations. So we have the packaging and packaging waste regulation, which really recognize the benefit of composting for some materials, and mainly when they are having introduced a higher bio-waste capture, so that's more bio-waste is going to composting, which is better than landfilling or burning our food waste, and also where there's a lower contamination of compost by non-biodegradable plastics. So this packaging waste regulation has made two types of products mandatory to be compostable. The first one is permeable tea and coffee or other beverage bags. So these are permeable and you can throw it away together with the tea and your coffee to the organic fraction. So it's logic that they are composted together. The other one example is the sticky labels, which is attached to fruit and vegetables, which is of course logic that they all would also be compostable. Nobody will remove these labels from your food before throwing it away. These are mandatory. That is possibly that also other things will be allowed, but it's depending on the member states, and this will be decided in the current year. So there are other products that can be allowed are non-permeable tea, coffee, or other beverage systems, single-use serve units. So that can be disposed together with the final product. Also, very lightweight plastic carry bags might be compostable, and other packaging that are already composted in these countries. But they should all be well underbuilt, and there should be also a composting infrastructure already in place. So really want to have dedicated products that can have a sense to be compostable. Next slide, please. So this is my last one. This mainly to show you also that we are really working on this in the ReBIOlution project. In this graph, you can see the biodegradation of three materials which were developed within ReBIOlution. These materials are bio-based, but can also be and are biodegradable under composting conditions. So as you remember, the requirement is that you have to achieve 90% biodegradation within 180 days. So our reference material is the cellulose, which quite went well. It passed the 70% pass level, and three products which are developed also showed them a very high biodegradation. So they showed more than 90% absolute or relative biodegradation. So these materials we developed in the project proved to be biodegradable in this compost conditions and are a good starting point to proceed within this project. Thank you for listening to me. Questions can be asked later. I'm now giving the floor to our expert, Sandra Mika, on soil degradation.
Yes, good morning. I hope you can hear me. It's a pleasure to meet you all online, and welcome to the seminar also from my side. My name, as Stephen already has mentioned, and Miriam is Michael Sander. I'm a professor here at ETH in environmental chemistry. I assume you all have heard of ETH or are familiar with ETH. It's a research university with a strong focus on science, engineering, and mathematics. It was founded about 170 years ago. And at ETH, it's a very international place. We have about 25,000 students from 120 countries and approximately 4,400 doctoral students. One or two or three of these, actually, I will present work of today in this webinar. They are the ones, together with the master students, that of course generate a lot of the science that we then have the pleasure to present. There is, as you will see, also from our part, but as you may have also heard from other research areas at ETH, a strong focus on sustainability and the environment. Next slide, please. What I have the pleasure of doing today is to take you underground, meaning I will talk about biodegradation of biodegradable polymers in soils. And similar to what Miriam has already alluded to, I want to give a brief motivation for why we are interested in biodegradable polymers, soil biodegradable polymers. On the left, you see a depiction that I copied from the Ellen MacArthur Foundation, essentially showing that we can consider two loops or two cycles in the plastic economy or also in material flow analysis. There's a technical cycle. This is the one we are well familiar with, in which we say we should reuse materials, recycle materials. But what is relevant to our presentation today and our discussion is a biological cycle, and that is an element cycle in the environment out in the biosphere. And so when we have polymers or plastics that deliberately enter the environment and are left in the environment, then ideally they should be part of this biological cycle, because that would ensure that they don't accumulate in the environment. Why is this particularly relevant for soils? Many polymer products have soil as a targeted receiving environment. Within ReBIOlution, for instance, we are talking about thin mulch films, which we now try to make out of new polymers, polyesters, and assess their biodegradability in soil. We have also seed coatings, geotextiles, fireworks, small pieces such as string mower strings that can enter the environment, and specifically in this case, soil environments. What is important here then is that these biodegradable polymers that are used in these applications are compatible with this biological cycle, and specifically, if we talk about soils, are soil biodegradable. Next slide, please. So when we talk about the process of biodegradation in soil, which steps are involved? Essentially, the polymer plastic item is transferred into soil, and in a first step, soil microorganisms, meaning both fungi and bacteria, start to colonize the polymer surface. This colonization will also happen on conventional persistent plastics. You may have heard of the term plastisphere. So this is not unique to biodegradable materials, but what is unique to biodegradable materials is the next step, that, for instance, extracellular enzymes are excreted from the microorganisms on the surface, and these extracellular enzymes then cut down extracellularly the polymer on which they sit into small pieces that are sufficiently small for microorganisms to take them up. And so eventually what you see in the lowest picture is that then microorganisms take up these little pieces that are liberated through the enzymes and then take them up and metabolize them to CO2 and microbial biomass. And what I will try to do in the following slides is give you an overview of research that we have conducted on this process, and I'm happy later on to discuss specific aspects if they're of interest to you. Next slide, please. So before I go into examples, I want to stress one point. I already alluded to it, and that is that these biodegradable materials are different from conventional materials in that they undergo this biological cycling, and in the end that is a microbiology question, but also a chemistry question of the polymer. And so one polymer I depicted here is polybutylene adipate coterephthalate, PBAT. It's fairly often used in biodegradable materials, and key to the biodegradation process are ester bonds. You can think of it, if you're not a chemist, akin to Lego, where you have breaking points between the individual bricks. These ester bonds are the places where enzymes can attack and then convert the macromolecule, the polymer, into small items, as you can see here, terephthalic acid, butanediol, and adipic acid, and these can then be taken up by microorganisms and converted to CO2 and biomass. In ReBIOlution, one of the major ideas is that we make bio-based, biodegradable polymers, and this is by including furan dicarboxylic acid, which replaces, for instance, a fossil-based terephthalic acid. Next slide, please. So I would now like to walk you through a couple of research results. I will not dive into too much detail, but essentially pointing out that these processes occur and that these materials biodegrade in soil. What you can see here are scanning electron microscopy images of PBAT after it was buried in soil for 6 weeks and then retrieved and then looked at under the microscope. And I hope you can see at this resolution that there are biological features on the surface. You can see in the top two images, for instance, fungal hyphae. You can also see unicellular organisms aligned with these fungal hyphae. And in the lower image, you can even see a direct image of biodegradation, if you want, and that the fungi is growing three-dimensionally into the surface of this PBAT material. Next slide, please. This is not only the case for PBAT. There's another polymer, polyhydroxyalkanoate, where you can also see biology colonizing the surface, and you can see around the biological features degradation due to enzymatic hydrolysis of the surface of this material. And so even the images already tell us that associated with the biological structures on the surface, we have degradation. Next slide, please. I will not talk in a lot of detail about the enzymatic hydrolysis part. We've done quite a bit on this for the ReBIOlution polyesters, and they hydrolyze quite rapidly. The point I would like to emphasize here is that in order to these materials to be biodegradable and feed into the biological cycle, they need to mimic existing structures in the environment. And one of these structures that the biodegradable polyesters mimic is cutin. You know cutin from the surface, for instance, of tomatoes, as you see here in the top left. And cutin is a waxy material, a biomacromolecule. And the point is it contains a lot of ester bonds. So nature has found a way to cleave down these polyesters, this cutin material, and it's essentially the same enzymes that are used for these biomacromolecules that naturally occur, in this case cutin, that also then cut down the polyesters. So the idea of biodegradable polymers in soils is always to make the structure of the polyester such that it fulfills its role and its function in the application, let's say a mulch film, but at the same time be similar to naturally occurring macromolecules such that enzymes that exist in the environment can cut down also the polyester. Next slide, please. What you see here that's a bit weird, looks funny. Sorry for that. You see a mineralization curve. So this would allude to the uptake of small products and the mineralization. On the left, you can see, if it was correctly shown, but you see the citation, polybutylene succinate, which is a biodegradable polyester. And what is shown is the mineralization of this polyester to CO2. My apologies for that. You can check the Nature Communications paper. This must be a transfer mech PC issue. My apologies. And on the right, you actually were supposed to see cellulose. Sorry for that. But the point is what I tried to show here, which is not showing up, is that these biodegradable polyesters not only are colonized and microbes are degraded, we also see that its carbon is being transferred to CO2, meaning that microbes actively metabolize constituents of these polyesters. What is specific about this experiment is that these are 13C labeled polyesters, such that we can clearly show in the CO2 that we measure, the mineralization product, that this is 13CO2, and therefore that the carbon, the 13C, has to originate from the polyesters and not from, let's say, soil organic matter. Next slide, please. We can also show that besides mineralization, that microorganisms take up part of this carbon from the polymer. This is again done with the 13C labeled polyester, in this case PBAT. And if you look at the left, you can see there is a technique called nanoscale secondary ion mass spectrometry. It's essentially a very accurate camera, if you so want, a microscope, that allows us to have nanometer-sized resolution on the surface. And what it does to us, it reports back to us the elements that we find on these nanometer scales. How is this done? You have a primary ion beam that you shoot onto the surface, and then if you have biological structures, for instance, you generate through this ion beam secondary ions, and these are then analyzed in a mass spectrometer. And so you can at any point on the surface determine which elements are present. If you then look on the right, you can see a secondary electron image, which is essentially a scanning electron microscopy image. You can see how the surface of the film is in the background, and you see biological structures growing on the surface. At the bottom, you can see which structures are actually biological, because you see 12C and 14N. These are ions that can only result when you image biomass, because the film PBAT does not have any nitrogen. So only nitrogen containing structures now show up. And essentially what we see highlighted are the biological molecule, the biomass, the microorganisms on the surface. Decisive for the conclusion of uptake is the picture on the right, where you can actually see and prove and demonstrate that the carbon that comes, the 13C, that comes from this specifically made PBAT is being incorporated into the microbial biomass. You can see that the biomass in parts has 13C atom percentages of about 5%. And we showed through control experiments that this is only possible when they grow on the carbon that comes from the polymer film, which you can see in the background to be labeled at 3.5%. It's the green area. Why is it even higher than the background? These organisms essentially accumulate the label in their biomass preferentially over unlabeled parts of the polymer. Next slide, please. This brings me to my conclusion. I hope I could show that and argue, Miriam also did this in the beginning, that soil biodegradable polymers are useful for specific applications and they should go into the biological cycle. Biodegradation is a multi-step process involving colonization, hydrolysis in the case of polyesters, and microbial metabolism, forming CO2 and biomass. There is strong evidence that soil biodegradable polyesters are actually fact, not fiction. Of course, the rates will depend strongly on which soil you look at and what the conditions are, but these materials indeed biodegrade. And coming forward will be hopefully soon papers that we are currently preparing on the biodegradability characteristics of ReBIOlution polyesters. Thanks for the attention, and sorry again for that one slide that didn't show up quite nicely. Thank you.
Thank you, Michi. So I take over to introduce you what we're doing in the aquatic environment. HYDRA is a research and test center, mainly focusing on testing on tailor-based projects, be it marine, freshwater, or soil. We also do some communication and visualization work and concept and consulting for this topic, be it in lab tests, simulation, mesocosm tests, or field tests. Next slide, please. In ReBIOlution, we are focusing on working pre-normatively developing environmentally relevant testing for the aquatic scenario, mainly focusing in the marine environment on the deep sea scenario, because for the shallow water scenario, we do have standard tests in place. And the question of the EU policy framework was, do we actually need deep sea test scenarios pictured or option to use for the policy or not? And this we picked up here in the ReBIOlution project. So we are looking deeper into that, how to establish a deep sea test and if that's actually needed. In the freshwater, on the other hand, we do not have a lot of standard tests available with environmental matrices, means sand and water from lake or river or big river scenarios. So we're looking into this to then actually propose a validated test methods for freshwater scenarios in a larger or end of the project that can give even some policy recommendations in this respect. Next slide, please. For the marine environment, I like to share some first results we have done with some sediment from the deep sea collected from a big ship from depths like 280 meters, 610 meters, and 3,330 meters. You see in the upper graph some biodegradability testing in closed vessels, so we can deduce the biodegradation. Here you see the results from our positive control. It worked really well. The sediment from the deep sea, be it from all the three places, are very active and are metabolizing the material exposed to them. In the lower picture, you see disintegration results where we deduce from then erosion rates, which we can then use in LCA. And you see here from 3 different sediment types, similar erosion rates. Some are a bit slower than others, some are more distinct. You see that on the high peak or to a rather broad peak. This reflecting then the security of our replicates or the data from the model deduced here by this experiment. What we also can say from this, we can assess the variability from different sediment points. And now, as a small outlook, is running a test under pressure, because we have to show the difference. And we would like to actually establish then, or suggest a rather simple test method, because this is quite labor intensive, what we're doing here during ReBIOlution. And so there will be more to come by the end of the project. Next slide, please. In respect of the freshwater environment, we are following up this scenario. A farmer is using a biodegradable mulch film here in this strawberry horticulture, then changing the culture, and some of the mulch film is now released into the environment, not really plot under, so it can actually be flushed into or by wind into the nearby waters. Also, this is depicted in the EU policy framework. Do we have to address this transfer of mulch film from soil to fresh water? You see here in the bucket what we collected in front of this tunnel after the change of the culture is quite a lot of mulch film, which could potentially end up in the nearby water. Next slide, please. So what we are doing here in the lower part, in the green cycle, you see the farm area and a small creek passing through, and a small lake nearby on the left side, and the creek then flowing into the River Rhine, potentially then to the North Sea in the shallow water, or even in the deep sea. So we go out and collect, or we went out and collected some sediment, and in our experiments, controlled tank experiments, we then exposed some mulch film equivalent on the sediment surface, so under oxic conditions, and then buried into the sediment, which also can happen, so in this case, without oxygen in the sediment present. Next slide, please. You see here the results of these experiments in the 3 different sediment types, one set up on the sediment surface and one in the sediment itself. Biodegradability was successfully assessed in the respirometer, and disintegration data you see here. And you see that they are all disintegrating in faster or lower rates. However, this is possible here to depict the variability, even showing under oxic and anoxic conditions. Important to understand here that these erosion rates are specific for the material and the environmental conditions. So we cannot extrapolate it to other materials or like other environmental conditions, like much colder or much warmer, or more nutrients and less nutrients. This has to be assessed in an extra test if that's of interest to know and to report. As an outlook, we will validate these tests. What we can say that they are working with environmental matrices. So most probably we can come up with some suggestions for standards and policy recommendations. Currently running some transfer tests from soil to freshwater. We don't really see, I can give a little teaser, not a real difference. Doesn't really matter if it was exposed to soil beforehand. However, we will see how this experiment will continue. By the end of the project, you will then get an update. Next slide, please. This is leading me to the end of our presentations. Here, a thank you to all the consortium. I think it's a really nice project from the point of view safe and sustainable by design, starting at the very beginning with new polyesters. And we can then also look already how they are behaving in the open environment and under composting conditions. With that, I'm leading us to the Q&A. Not much time left, I see. However, maybe you have some questions, and I'm also happy to stay 5 minutes more. And certainly you can reach out to us by email.
Thank you, everyone, for bearing with us during the entire presentations. We had some technical issues at the beginning, but hopefully it will be solved after we upload the full presentation on the YouTube channel. Now, passing on to the questions, we have some questions here for for our speakers. I'm gonna say them out loud, and please, you guys answer. Okay. So the first question by Martin, he was asking, can you comment on which EU policies or regulations target home composting and why that would make sense? I think it's directed to Stephen, probably.
There is policies. There's no policy directly on industrial composting. The packaging and packaging waste regulation mentioned something on home composting that some countries can decide that it should not be only industrial composting, but also home composting. That's included. So that's what member states are now discussing and setting up. But for some application, home composting can make sense because it's if you can make it home composting, it's by far also industrial composting, and it's not all facilities or all environments have a separate collection of organic waste. Certainly not in the more rural parts of the country. So that's why home composting can be a nice alternative.
May I add here that I'm always raising the flag reduction of pollution. So this is always a benefit or an added value. And I think it would make a lot of sense that the materials which are compostable also are environmental biodegradable in case they are ending up in soil or aquatic environment.
Thank you. Next question is from Rick. He was asking on slide 29, how do you know that labeled C does end up in microorganism as a C building block of said microorganism and not as a fragment of nanoplastic inside of the microorganism?
Yeah, I think this
that's
question
a
goes
question
to
for
me.
Sander. Yes,
thank you.
exactly.
So thanks for that question. It's a very good question. What we see obviously in this experiment that we ran is that there's mineralization to CO2, and it is well established and known from naturally occurring biopolymers that when microbes utilize substrate, part of it goes into respiration, CO2, and part is always used to repair or build new biomass. And so it is not surprising that we find some of the carbon also being incorporated into microbial biomass. How can we delineate whether this is pieces of plastics, so to speak, or biomass newly built? It's very clear in this case that it has to be biomass newly built. The particles would be too large to take up. Only oligomers and monomers can be taken up across the cell membrane. This is not typically seen for larger materials. The second is that you saw the isotopic composition. It was even enriched. And this is only possible in this case if there is a rebuilding using the monomeric unit that we labeled to build new biomass. So this is taken together very strong evidence that this is carbon that was directly incorporated and not particles being taken up. I hope that answers the question.
Okay, thank you very much. I was going to ask the one from the chat, but I see that Sander has already answered it. So I won't ask that one out loud, but we have another one from Rick also. He's asking, at EC level, how is the notion of biodegradation in relation to persistency? Is there policy or drive to promote materials that are not persistent but do not pass certain composting biodegradation tests because they are slower degrading? So I think that was for Miriam in this case, I think.
Okay, thank you for this question. Yes, when we do exchange with European commissioners, we do get a positive response, especially for applications which have to last longer than a short lifetime. We do have to come up with other solutions than the current test standards available for like composting, which are supposed to have a certain time frame, because the composting facility is not running for ever, let's say. And the issue is rather here to come up with good test methods and suggestions, because then a slower biodegradation will mean a longer test time. And this is productive for the development of new materials, and then also for the testing and then policy implementation. So this is clearly some work to do on the research side here.
Thank you very much. A couple more questions. We have another one from Rick, who was very active in the questions. He says, EC frequently refers to the lack of EC accepted standards for biodegradable materials. While there are many test standards, etc., what's the issue precisely here and what to do about it?
Maybe I can answer here too, is what I learned, that some of the ISO standards available for example, in the marine environment are not yet on EN level. And why is this important? Because some thresholds, especially for metal content, as Stephen explained in the flow chart on the EN for industrial composting, have to fulfill a certain threshold, which is lower on EU level. So you have to adapt the ISO standards to EU level, or so it's a EN ISO, and this has to be done. It is not happening just like this. It has to be done by a working group.
Thank you. Well, the final question is from Angus. It's for Stephen. It says, would you recommend that all biodegradation tests for comparison of polymers are performed at the temperature of 28 Celsius for standardization and to relate to EU regulations?
He's now mainly referring to the home compost condition. I think for me, not. You really have to set your temperature in relation with the environment. Some polymers which are now commonly used as industrial degradable polymers, will not degrade at a sufficient quick rate at 28 degrees. So that's logic that you, for industrial composting, go to a higher temperature of 58, because you have long periods with really high temperatures. And in fact, for soil conditions or water, these 28 degrees might even be quite high to be realistic. Of course, if you only want to see to really have an optimized test with a fast result, then it can be a good temperature also for this organism. But for me, you have to relate it to the environment.
Okay, thank you very much. I think that sums it up for us. And I want to thank you on behalf of the entire team for being with us the entire process. And I hope this series of webinars has been very helpful to you all. I don't know if you guys want to say anything else, just a quick goodbye, okay?
Thank you to all. Don't miss the next webinar and join us on the website or social media. And
Thank you very much.
Thank you very much for joining us.
Thank you. Bye-bye.
Bye-bye.
Thank you. Bye. Listo.